How's it going?
Yeah, not too bad, not too bad.
Just
Yeah. How are you doing?
yeah, good. Just a little audio check. Do I still sound okay from yesterday?
Yeah.
Yeah, okay, wicked. Hey Arthur. This is Arthur. So he does the same course that I do.
Okay.
And he's doing some really interesting research actually on, well, I'll let you explain. Cool things with manganese and microbes.
Oh, wow. Amazing. Hey, Peter.
Hello.
Hey.
Hello. Good to see you.
And you. I might, if I can extract some sensibly priced EM1 out of Martin, Tom, I'm going to get in touch with you to see if you might want some.
Okay, yeah. You definitely want some. I've also got a couple of liters, Peter, that I haven't had time to use yet. I want to. We need to. So yeah.
I haven't, to be honest, Martin. I've only just got back to my emails and I saw messages, and I saw that you got back to me, and I haven't read the amount, except that you did say you only had 10 liters, and I was thinking, oh, blimey, that could be lots. But anyway, cool.
1 litre should be back in stock on the website. Have they not shown?
Oh, brilliant.
Yeah, so 1 litres are back in stock now. But for your project, 100 mil will do 20 kilos or maybe.
I know. So I don't really want 10 liters. I'm looking at it and thinking,
absolutely
no. But thanks. Okay, I'll get back to you, Martin, the next tomorrow or the next day. Thanks.
Awesome.
Yeah.
Lovely stuff. Hi, Joanna. Hi, welcome.
Hello.
Hello. Okay. Just give everyone a couple of minutes to arrive. So Arthur, what are you studying the same course as Mary as well?
Yeah, I am.
Okay.
Can you hear me?
Yes, yeah, we can.
Yeah, just doing the same, like part-time. I'm just a year earlier than Mary.
Okay, cool. Can you remind everyone what that course is?
It's called Soils and Sustainability at SRUC in Edinburgh. So it's at SRUC, really, the Scottish Agricultural College, but we kind of have all the resources of Edinburgh to it's very broad and open to kind of explore whatever we want, which is really nice.
Yeah, awesome.
Yeah, I thought it was like a really niche course going to. I was like, yeah, I'm finally like honing in on this really niche topic, and I realized like soils are basically like the ocean of the land, and so there's so many different things that are covered in that course and directions you can go in.
So Mary, would you mind if I read a passage from a book that I really felt was relevant to
Oh my God, I would love that. Please go ahead.
Thanks. So I've been reading this book, guys, called We Are Nature Defending Itself. It's fascinating account of like activism in France, actually, at this place called Les Ades, which is this huge area, like 3,000 acres of wetland, which has been defended by the people of that area in northern France for like several decades now. And it's been the host of some incredible stories, but basically it's a real joining up of like activists from all over the world with like the farmers and the people on the ground. And the police have they've described it as this communist thing that must be crushed, but actually it's some incredibly connective and powerful resistance to the sort of state capitalism, basically. But there's just this beautiful bit in it that I wanted to read because Mary is a self-described mycophile, which I think is beautiful, like a lover of mycelium.
I'm back.
Oh,
so,
okay.
Internet decided to cut me out. I was being too subversive, maybe. I don't. So this is chapter, because this book is all about it's about art and activism, and it's really good. But I'll just read this chapter because I thought it was so relevant to tonight. So, okay. How we understand life itself will decide our future. The classical models of biology, a little under 200 years old, created the conceptual framework that enabled us to turn the world into a resource. But this blindness toward what life is being shaken to the core. In fact, The revolution is profound as that of a century ago, when relativity and quantum theory reframed Newtonian physics, now biology, with its ability to sequence genes, to look deep into the microcosmic world, correlate huge data sets, and understand the entire genome of a being, is showing us what so much traditional and indigenous knowledge knew all along. Life is not a machine made up of discrete building blocks, but a dynamic, interpenetrating, interrelating whole. It does not calculate, but feels. It is not dead matter, but alive and expressive at every scale. Something that the Cartesian worldview sought to purge from human understandings of life is returning. It seems that we can only understand living beings if we reintroduce subjectivity. Experiments show that when two genetically identical seeds are put into identical settings, each one can grow very differently. Each one is a self, committed to its ongoing existence in its particular body and embedded in and interpreting a particular place. As their roots emerge from the seed, there is nothing deterministic going on. They are not just following orders from efficiency-obsessed DNA, nor do they just fumble about randomly. They choose to head toward what will enable the plane as a subject that interprets external stimuli and genetic influences rather than being causally governed by them. We could imagine DNA as like a musical partition interpreted by musicians. Organisms seem to act according to values and create meaning because they sense the world. This is not necessarily about cell consciousness, but we could view this as intelligence. The Latin verb intelligere means to be able to discern between things, the ability to choose existence, to experience a concern for a core self and express it is the very definition of life. If feeling is a physical force and the expression of this feeling is a physical reality whose meaning motivates organisms to act, writes Andreas Weber, then we might understand living beings better if we imagine what is happening in the biosphere as, in a way, resembling artistic expression. Art, then, is no longer what separates humans from nature, but rather it is life's voice fully in us. I thought that was really nice.
Oh man, that was beautiful. I definitely think fungi help us demonstrate that as well, for a fact.
Yeah. So your work with like mycelia and stuff, and I know there's a whole crew of you doing that up in Edinburgh area, is so cool as well. I know that
yeah, I think I love what I love about mycology so much is like it's such an accessible subject. Like doing the Mycology Society here, like we really are trying to like bridge art and mycology, and it's making us think more creatively in the way that we need to like be able to understand these kind of dynamic relationships that are going on. So yeah, that was beautiful though.
Thanks. Great. Wicked. Well, excellent. There you go. You're the host, Mary.
Okay, right. Nice one. Okay. Can you see my screen and the slide?
Yes, we got it.
Yeah. Okay, wicked. Let me move these Zoom controls out the way. Okay. So hello everyone, mycelial beings. Yeah, firstly, I just wanted to express my gratitude really for you all joining me here today. And I just wanted to shout out to Tom and everyone who's helped set up this network really, because you know, it's really awesome to be part of this growing hive mind of inspiring beings and projects. So yeah, I'm really digging it. So let me then take a moment to share a bit about my journey. So I didn't actually have a background in science, but fell deeply in love with soils over the last few years, which kind of just ended up with me nerding out really hard on soil science. So now I'm doing a master's in soils and sustainability at Edinburgh, but joint with the Scotland's Rural College. However, I'd also say, and it's kind of been sort of said here, my real passion as well is around fungi. So I also run the Mycology Society here where, you know, alongside my soil endeavors, I spend a lot of my time engaging people in the fascinating world of fungi. And And generally, I would say that I'm a big advocate for education, so especially like soil education, sharing knowledge and making information like digestible and engaging, because, you know, soils are incredible and important. And, you know, I think, you know, the more that we can help each other to understand and, you know, share knowledge, the more that we can help each other to look after them better. And I definitely think like the really complex and dynamic nature of soils as well, it really requires these like collaborative conversations between each other, because, you know, all of us, we're like these, you know, we all hold these like individual unique pieces of the puzzle, right, to understanding it, you know, whether that's you're coming from a science or a research background, or you're like directly engaging and working with the land and the soil. Like, something I really like about this network is we all come from really diverse backgrounds, right? So it's just really great to have this like cohesion of knowledge sharing together. And on that, oh, another thing I'll mention is, yeah, so I've started doing a bit of content creation as well. So this is mostly through my Instagram, soils and fungi. And the main thing here is just, you know, I like to consolidate information on soils and fungi, really, which basically just kind of helps me learn, because, you know, the best way to learn is to teach. And so I decided to kind of take this a step further then and consolidate my learnings on soil organic carbon. And so carbon, it's not actually something that I thought I would ever get that interested in, but I ended up getting really absorbed into it since doing my course. And, you know, kind of like when you start learning a lot of things about soils, you just kind of end up feel like you sort of opening up this fractal that kind of just keeps infinitely expanding with complexity. And I just thought, you know, given the global relevance of carbon now, like I really just wanted a space to share an appreciation for how complex the cycle of soil organic carbon is. And hopefully I can plant some like seeds of insight and, you know, maybe spark some good discussion on this. Like, I would really love to hear what you guys think about any of the themes that I've covered, you know, maybe bring in your own interpretation as compost creators, for example. There we go. So here's a little rundown of the journey that we're going to take. So I'm going to first of all cover what I mean by soil organic carbon. And then I'm going to cover, you know, why it's important, the implications, global relevance. And then I'm going to focus on the key paradigm shifts which have occurred in sort of soil science and science due to our understanding on soil organic matter. And then I'm going to mostly focus on the key sort of stabilization mechanisms behind the sort of protection and stability of carbon in our soil. And then I'll finish by showing you guys a really nice way to measure aggregate stability in the field and how this can actually serve as a potential kind of proxy for measuring soil organic carbon. And hopefully we'll have some nice time for questions and discussion. So what is soil organic carbon then? Well, what I'm talking about is the main component of organic matter. So this nice little slice over here of this lovely pie diagram, I'm sure we all recognize. You know, we aim for the soil to be around 5% organic matter. And so around 55 to 60% of this is carbon by mass. And so this kind of encompasses, you know, like a continuum of fresh to decaying plants, microbial, soil faunal, debris and exudates and stuff. And, you know, a lot of this carbon is then primarily derived from photosynthesis. So our autotrophic organisms like plants, you know, will fix some of this carbon into, you know, more available forms, which then end up cycling through heterotrophic organisms, so like microbes, who will then, you know, utilize this for energy and can break it down further. And this whole thing, you know, it's a cycle. So at the same time, you know, a lot of this carbon is then being simultaneously released back into the atmosphere as CO2, carbon dioxide, where, you know, it's respired by various microorganisms, but also partially by plants and other meso and macroorganisms as well. So I know there's a lot going on here, but I just wanted to show you guys the kind of diverse forms that soil carbon sort of takes. So a lot of carbon, you know, it's found in our plant inputs. So, you know, a lot of this carbon is in plant biomass, their decaying plant litter, and, you know, very importantly, their roots and their root exudates, where, you know, this is where most of the carbon is actually entering the soil from. And so a lot of this includes like simple sugars, so particularly made out of glucose, which is kind of like the basic building block for most of the molecules of life. And then we've got things like organic acids, amino acids, proteins, and these are kind of examples of metabolic compounds. So these are the things that really help the organisms to convert energy into nutrients, and also help to break down other molecules as well. And so once in the soil, lots of these carbon inputs, they're obviously also undergoing various stages of decomposition and microbial processing. So, our hungry microbes play a crucial role in assimilating some of this carbon and then producing their own set of organic compounds as well. And as well as that, a lot of organisms have, the stuff that they're made of is made of these different structural compounds. So, one example is polysaccharides. So polysaccharides are basically just like a carbohydrate, so like a chain of sugars. So fungi, for example, they've got something called chitin. So chitin is a polysaccharide. So this is what makes up their really rigid, strong structure that's found in their cell walls. And plants, they have similar. So plants have polysaccharides like cellulose. And they also have other important structural components like lignin and lipids and stuff. And also, a lot of this carbon is remaining in dead organisms too. So like dead microbes, that's often what we term as necromass. And this has become quite a hot topic of research now and something that I'm going to cover later in this presentation. So why am I talking about soil organic carbon then? Well, carbon, it's extremely relevant now, where it's embedded in our environmental, our political, our economic and scientific spheres. Like we really do live in this sort of carbon dominated era. Carbon, it's currency now, like carbon markets and the monetization of carbon, like, it's kind of moving in some quite wild ways as well. Like we're hearing more stuff like this. So farms, like quite a few in Wales, for example, are sort of being bought out by these intermediary companies to offset carbon emissions from other companies somewhere else by planting a load of trees instead. And I just wanted to use this as an example of how, a lot of our focus on carbon is dictating these shifts in capital, land use, livelihoods, decision-making. And like, this is like one element, right, in its various states that we're like kind of capitalizing and grasping at, but there are still so many existing uncertainties, and you'll kind of see that as I go on through this presentation. So obviously a lot of this has stemmed from our efforts to mitigate climate change, right, where the spotlight has shifted towards the roles that soils can play in carbon sequestration. So we've all heard this analogy of like, the soil being a sink or this reservoir with untapped potential to store more carbon. But unfortunately, it is a double-edged sword when it comes to soil carbon, because, the way that we manage our soils, it might be able to turn it into a net sink, but equally it can turn it into a net source of greenhouse gas emissions. And, I'm not just talking about CO2 here, I'm talking about methane, nitrous oxide, these potentially even more serious greenhouse gases that are far less talked about. And, kind of personally, like, while it may kind of be less glamorous and attract less media attention, like we really should be focusing on protecting the largest terrestrial store of carbon on the planet, because, and rather than portraying the soil as a remedy to our own emissions. And so what I'm trying to get at here is the significance of this topic. Like, it can't be overstated. And ultimately what I want to really emphasize throughout this presentation is that the fate of soil organic carbon is far from straightforward, where its cycle, its stability, its residence time, this remains the subject of intense research and debate, which really kind of transcends any kind of random analogies or simplified perceptions that have kind of been driving the ideas around the carbon sequestration potential of soils. But I do quite like this quote from this paper, which describes soil organic carbon not as this trapped reservoir, but as a stream of carbon atoms flowing through the soil, propelled relentlessly by thermodynamics to higher entropy of CO2. And what this is basically saying is this, the carbon is in flux. It's continuous. Like, it isn't in its nature to just stop and remain fixed at a point. And instead, like, any temporary arrest is, influenced by a complex range of biological, chemical, and physical mechanisms. So how then has our understanding of soil organic carbon evolved, and where is it currently at? So in the past decade or so, there's been some key paradigm shifts within science and soil science to do with our understanding on the fate and turnover of soil carbon. And this is very much nested within the realms of like trying to understand soil organic matter and decomposition of matter generally. And this has been kind of driven by, advances in technology and analytical methods. And sort of for carbon, the key focus is trying to understand its residence time. So, how long is it actually staying there in the soil? Because, this is what has the implications for climate change, right? But these kind of emerging findings and, ideas around its residence time, this is what has caused quite a lot of divide in the scientific community, where theories and research are still, very much evolving. So this paper, published in 2015, it's quite popular, kind of for its controversial nature, which sort of thematically upends traditional soil science and scientific theory on organic matter and decomposition. So this is sort of the general theme. So it opposes traditional humification theory, which is actually kind of just based on this historical method which uses alkaline extraction to isolate and measure the fraction of soil organic matter, where this, demonstrates the existence of a humification process, which is how, organic matter is shows how organic matter is then decomposed and transformed into these sort of humic substances. So it assumes this creation of these large and recalcitrant humic substances. And so this went on to then assume that these had these unique molecular properties which made them resistant to microbial breakdown. And this makes up the majority of what we've always called soil humus, and what we've always believed is like this kind of stable and inert fraction of the soil. Now, this paper, it proposes quite a few things, a new model called the soil continuum model, but I'm not going to get too much into that, but I'm more just using it as an example for the shift in narrative in science now, where we're now starting to conceptualize soil organic matter not as this chemically defined or stable or inert fraction of the soil, but rather as this continuum of turnover times, which is moderated by the physical constraints on the accessibility of organisms and their extracellular enzymes. And to put this more simply, if soil microbes, if they can access soil carbon, they can mostly degrade it pretty rapidly, almost regardless of what its molecular structure is. And so, this kind of makes sense as well, right? Because, we're always hearing these microbial success stories of fungi breaking down, really complex contaminants like hydrocarbons. So therefore, research and theory on sort of soil carbon dynamics now is more focusing on these biologically non-preferred soil spaces where the soil carbon is effectively being physically protected from microbial activity. But before I move on here, I just wanted to say, we can't actually completely outrule some degree of molecular resistance to microbial breakdown. There are some examples. So you might see these hydrophobic kind of coatings of lipids on soil particles, for example. So that would be kind of an exception there. And what I also wanted to mention is despite any of these new developments, we are still very far from a unified theory or understanding on soil organic matter and therefore carbon dynamics. And, I think we can all agree, the soil is an intricate universe. And I actually sometimes think that it would take maybe an entire shift in our conscious or perceptive capabilities to really be able to grasp its dynamic nature, kind of a bit similar to what Tom was saying in that passage he read out of. That's probably the kind of mindset we need to be working with here. But this is what also makes it so exciting, right? And nevertheless, we can actually agree on some key features. So here's where we are now. We've shifted the focus away from assuming this like chemical resistance or recalcitrance or inherent molecular stability of soil carbon. And instead, contemporary research efforts are now more emphasizing these sort of key stabilization mechanisms, where sort of carbon storage is now being focused on, these sort of wider ecosystem properties. But one thing I will mention is, so some of these are like mineral adsorption, occlusion with aggregates, microbial ecology, and necromass. I'm going to go through some of these, but I just wanted to point out that while the emphasis has shifted to here, like this is ultimately still all being governed by these key controls over here, temperature, moisture, redox, pH, and other factors such as soil depth and soil type, which are really important. And something that's quite funny in like soil science is like soil scientists, they've been working really hard, some of them, to like try and find like this holy grail of like the one soil property that has contributed to carbon storage. And really there isn't one thing. Like it's just a multitude of factors that varies across a multitude of ecosystems and biomes. So one thing that that's historically kind of made research on soil organic matter and carbon difficult is because organic compounds often adhere and bind to soil minerals. But this is actually one of the mechanisms that kind of protects it from microbial decomposition. And this adsorption of soil carbon or organic molecules sort of onto these mineral surfaces, like this is a really like complex and dynamic and intimate process of association, which, can involve clay minerals, but what's been more emphasized now is these kind of important metal oxides, so things like iron and aluminium, and I think manganese as well. And this is largely explained by the relatively large and charged surface areas of these mineral sort of sites. Now, there are various mechanisms, behind the binding and chemical wizardry going on here, which I'm not going to get into. Also, I definitely it's a bit beyond me that for now. But what I wanted to emphasize here is that the main thing that's driving the temporary arrest of carbon here is that the adsorption capacity or association is exceeding that of the localized enzyme capacity in microbes that are in that site. And this has been found to explain, the longer residence time of, this kind of carbon in the soil. But how stable are these mineral complexes then? Because, so I read a paper recently, which is quite interesting, and it highlighted the fact that these mineral complexes, so, with this adsorbed carbon, it can be dissolved microbially under anoxic conditions. So, in the absence of oxygen. And so these organo-mineral associations, these typically form under aerobic conditions, and so in the presence of oxygen. And these can actually be lost when the soil becomes anaerobic. And this can happen because they can sort of be dissolved by what's called microbial dissimilatory reductions. So by these kind of specialized anaerobic metal-reducing organisms, which include both bacteria and archaea. And they can essentially liberate this soil carbon. And so when the soil then becomes aerobic again, this mobilized carbon can then be quickly oxidized by other microbes and respired as CO2. So imagine the implications of this, right? Because, the soil, it's constantly moving through these periods of drying and wetting, aerobic and anaerobic states. Like, I know a lot of you composters out there, you're probably quite well versed with this dynamic by now. And which is even happening at this microscopic level in between all these little pore spaces. So when I read this, I was like, damn, do you know what? I've totally been neglecting the air and the water again, the stuff that makes up half of the soil, like aerobic and anaerobic cycles. And this is a really understudied area, and it's clearly an area that needs a lot more research, especially if we think about things like climate change, where, the extremes of drying and wetting events, they're only going to increase. So I want to move on now to microbial ecology and necromass. But before I dive in, I just wanted to actually emphasize the importance of this topic. And this is because up until now, carbon and climate models, the things that we use to predict climate change, they haven't explicitly involved microbes enough, like in terms of how they affect carbon storage. And like, this is mad if you think about it. And so thankfully, we are starting to see like a bit more emphasis now on microbial inputs. So, you guys, living soil and compost creators, like, really shifting the focus of inputs and amendments onto the actual soil microbiology. Like, this is really important stuff. So I'm hoping that we're going to be seeing this, more microbial focused carbon and soil management. Because, ultimately, to understand soil carbon dynamics, it really does rely on this understanding of microbial processes where soil carbon, it's dictated by this balance between microbial activity and carbon inputs. And, the relationship between microbes and soil carbon, it's almost kind of paradoxical where, they not only can reduce soil organic carbon stocks by mineralizing it and then respiring it as carbon dioxide, but in turn, they can increase the stability of soil carbon through their biomass and their residues. And with this, we're seeing a greater emphasis now on microbial necromass. So here's a lovely little diagram, which is kind of reflecting microbial necromass in the wider carbon cycle. So for example, you've got like your inputs coming in here from your plants, so your plant biomass, roots, root exudates and stuff. And this is kind of stimulating this pool of microbial biomass here. So obviously the microbes, will partially mineralize some of this, respiring it as CO2. But then, the rest of it is being stored in their biomass and their residues. So when the microbe dies, their remains are left in the soil. So their intact cells, their extracellular material, leftover bits of their enzymes, and, anything that kind of survived being devoured by the next microbial generation. Like, this all forms like this pool of microbial necromass here. And this obviously contains, the various organic compounds and carbon containing, components. And so then what happens is some of these fragments of necromass, these can then be stabilized further, where they'll be, either occluded within soil aggregates or will be binded and adhere onto soil mineral surfaces. And, so this is just something that I went through. So this pathway of microbial necromass then especially into these sort of protective formations, this is a really interesting and increasing area of research. And Martin actually shared a great paper on the network recently, which looked at the turnover of bacterial biomass carbon, which is then found to sort of partially incorporate further into fungal biomass. And so this is a really interesting area of research, which is growing, which is sort of looking at the relative contributions of bacterial and fungal biomass on carbon stabilization. So a lot of these studies, what they're doing is they're using stable isotope analysis of amino sugars and acids. Essentially, this means that they can just track this like stable carbon throughout these different like microbial metabolic pathways, because different microbes, they produce different amino sugars, for example. So, fungi, they produce like the majority of glucomazine, for example. And a lot of this stuff, this is what makes up like their cell structures and their their structural components, which is obviously then can persist as necromass after death. But what I kind of wanted to mention here as well is that, regardless of studying the relative contribution of fungal or bacterial necromass, like both of these have been shown to significantly decrease, obviously, with land use intensity. So this is something that we've really got to bear in mind. I mean, I think, most of us are aware with like disturbing the soil, we're breaking up those, key fungal networks, but as well as that, we're disrupting this sort of necromass pathway of carbon stabilization. So keeping slightly on the topic of fungi and microbial ecology, I thought I would mention mycorrhizal fungi, and also because I can't go without mentioning my favorite organisms ever. And also because this is a area of research which I'm quite interested in pursuing. So I'm quite interested in maybe looking at the influence of mycorrhizal fungi on carbon and nitrogen cycling. And, maybe looking at this sort of in the context of reforestation. So, all the surge in tree planting that we're seeing, it hasn't really been met with this consideration for the wider ecological consequences, right? And, that includes our fungal communities. But what's really interesting is this research and observed variations in how soils are storing and cycling carbon and nitrogen, depending on whether they are dominated by trees, which primarily associate with either of these mycorrhizal types. So just to show you guys, we got the two mycorrhizal types. We've got endomycorrhizal fungi. This primarily what we know as arbuscular mycorrhizal fungi. And just a side note here, these are some of my favorite fungi. Never even seen them, but, because they spend most of their lives living so intimately in the root cells of plants and the soil. And then, of course, we have ectomycorrhizal fungi, and some of these do actually produce fabulous mushrooms, like our classic fly agaric over here. So they're a bit easier to tease out. But what I wanted to show here really was an example of how mycorrhizal types can differ in their effects on organic matter formations, where these plant and fungal associations can actually influence the distribution and concentrations of carbon in either particulate or mineral associated organic matter forms. So just to reiterate here, particulate organic matter, sort of free living in the soil, whatever. This is generally recognized as the more labile and accessible of the organic matter, whereas the mineral associated organic matter, this is, our organic molecules are binded and adhere to these soil minerals, as I just touched on. And so a very fascinating study which came out recently, which I think I've put in the reference section at the end here, tested how soil factors, climate, and plant and fungal associations influenced the distribution and concentrations of carbon and nitrogen as well in these mineral associated and particulate formations. So this study was conducted in the temperate forests across the United States. And one of the findings which echoes a lot of other research is that forests dominated by AM fungal trees, so this is our arbuscular mycorrhizal fungi, these actually showed higher proportions of soil carbon stored in this mineral fraction. So this is really interesting, right? Because as I've just gone through, the carbon that then, eventually makes its way to be binded onto soil minerals, this is what is prolonging its residence time in the soil. Though it's kind of worth bearing in mind, though, is that, a lot of this can vary across different climates and biomes. So what I found is, since looking a lot into mycorrhizal fungi, some of it can be quite context specific, but generally what I've noticed is there are these definitive balancing acts happening between our plant and fungal communities, that I think really needs to be considered in restoration efforts, especially if we're thinking about carbon as well. But this is actually kind of starting to happen. So we're seeing more projects like this. So things like fungi, they're sort of doing these scaled up fungi driven restoration projects as part of wider reforestation projects. And so they're sort of using like DNA sequencing and machine learning to kind of try and identify the right types of fungi to be restored in different locations. But what's interesting is that part of their plan is to then measure some of the carbon that's been captured or stored or increased as a result of this, and share this information with corporations seeking carbon credits. So, this is a really quite niche example of carbon credit schemes in action now. And what's interesting is I noticed they're similar being implemented here in the UK. So I don't know if some of you guys have seen this, but there's the landmark carbon study, which is by the carbon community. And so they're trialing like these plantations of trees. So they're doing two main trials in Wales, doing two sort of main forest types. So one is a mixed broadleaf. So if we think mixed broadleaf, I'm thinking like, okay, well, that's going to have like your sort of diverse different mycorrhizal types, right? And then their other trial is a standard conifer monoculture. So when I think of that, I think, yeah, that's primarily going to be like just ecto mycorrhizal fungi, if anything. And they're also comparing this to like a control to regenerate naturally. And so some of these trees are being actually planted with soil that has been taken and transplanted from other nearby native forests. So, and I think some of them are also being given like commercial mycorrhizal inoculants. So yeah, I think this is really interesting. I'm really interested to see what the outcomes of these projects will be. Like, just thinking about the effects of different tree species and mycorrhizal types on, the effects on carbon storage. But I also think it's a really interesting area, like just in terms of thinking about microbial restoration, like, which is still a very uncertain area, like, considering that we've like barely scratched the surface in terms of species identification, let alone roles and functionality. And I also wanted to squeeze in talking about soil depth, mainly because, over 50% of global soil organic carbon is actually located in these deeper layers of the soil. So this is specifically below a depth of approximately 20 centimeters. And so some of this carbon, it's estimated to be super old, with like radiocarbon ages of like, between 1,000 and 10,000 years old. And this has been linked to sort of greater mineral protection and physical protection at these depths, also because when you get this low, it's obviously quite environmentally constraining for microbes. So they're obviously got smaller communities. There's not really many of them there. And so it was initially thought that this carbon, though, was relatively stable, but more studies are indicating that this deeper and older soil carbon, like this may actually be more susceptible to rapid decomposition than previously thought, like especially by human activity. So, there's obviously a lot of research now on the effects of deep plowing and tillage on the subsoil layers. But despite this awareness, the majority of studies on soil carbon and, soil sampling generally, we tend to only sample from like 20 centimeters to 30 centimeters. So that's all we've mostly ever really done. And so this obviously creates like a huge limitation and really like a lot of uncertainty in, global soil carbon estimates and climate models and stuff. And, generally our understanding of soil carbon cycling generally, like, so clearly more research is needed, which involves deeper soil sampling, right? Oh, I've skipped too long. There we go. So I wanted to finish up with talking about aggregates. So occlusion within aggregates is well known to provide sort of protection for soil carbon by making it essentially makes it like inaccessible to enzyme attack and generally like limits the activity of microbes by like environmental constraints, for example. So like an inadequate supply of oxygen, especially if you're thinking in these tiny little pore spaces. And to provide some context here as well, here's a diagram of a macro aggregate. I'm kind of sad actually, because I had been drawing my own diagram of a macro aggregate to include here, but I didn't get it finished in time. But this one will do. So we sort of this is a macro aggregate, and we sort of arbitrarily define this as being about 0.6 millimeters in diameter. And this is then made up of lots of little smaller aggregates, which we know as microaggregates, so these little ones here. And it's these microaggregates then that offer the greatest protection for soil organic carbon, because they've got these incredibly tiny pore spaces, which most microbes and their cells are bigger. So like, I can't remember the size now. I think it's like less than a micrometer or something like that, where there's also this kind of inverse relationship between aggregate size and the energy that the microbe would actually need to disrupt it and to like get that soil carbon that's in there. And this whole aggregate, it's held together by the adhesive properties of soil organic carbon. So you've got your microbial exudates, your root exudates, you know, your secretions and excretions by soil invertebrates. Like I realize I'm so biased for microbes, but like, you know, our earthworms, our unsung heroes, you know, really contribute to this aggregate formation process, and all of this lovely organisms gluing matter and stuff. This all strongly kind of absorbs then to these negatively charged soil particles. So all these gray dots you see here, these are clay particles. So they absorb quite strongly to them. And all of this is further enmeshed together by fungi and roots and stuff. And I really think actually that aggregate formation, I actually think it's one of my favorite soil processes, because you can like hold a little aggregate in your hand, and they're just like these mad, like multi-dimensional complexes that really kind of involve like physics, chemistry, and biology. So yeah, I love a good aggregate. But anyway, there is also a trade-off, which, you know, to actually maintain, you know, the stability of this aggregate, this depends on the turnover of soil carbon in here, where, you know, the microbes that are contributing to the, you know, the adhesion of this aggregate together, you know, they need that carbon. So if the supply of carbon is too restricted, then the stability of this aggregate is going to end up declining. So given the importance of aggregate stability, then here's a nice and simple method for measuring the stability of aggregates in water, which is known as the Slake test. Now, I'm a big fan of like anything that's like citizen science, accessible methods, especially ones where you're actually like getting your hands in the soil and dirty. And this was a really good one for that. And it's also a really great indicator generally of soil health. So I'll briefly go through what you would do here. So you'll get your soil samples. So I don't know, get a spade, dig a spade depth of soil out, and then, you know, break some of this up with your hands and try and locate the aggregates. So, you know, the little blocks that are measuring about, you know, roughly about 0.6 millimeters. Arrange this on a sieve, immerse the sieve in a bowl of water, leave it there for 5 minutes, set a timer, and just watch what happens. And at the end, give the sieve, move it up and down a bit, and just observe the remaining aggregate contents there. And what you can do then is then translate that to some scoring. Oh, what have I done? There we go. Then you can refer that to some scoring criteria. So I've just pasted this in from a research paper, but this is the kind of scoring criteria. So it goes from like 0 to 8, where 8 is like premium aggregate stability. Like pretty much all of your aggregates are kind of sort of still sitting there in the sieve, and like barely any of the soil is sort of invisible in the water. Whereas, you know, this gradually declines when you've basically lost most of your structural integrity of the aggregates, and the whole bowl is just looking pretty brown. So it's quite fun when we did this experiment. So we did an experiment we did like on a farm, we looked at 3 different land uses. So an arable plot, grassland, woodland. Obviously the arable plot is the intensively managed bit of land. So when we put our sample in there, it's just like, it was pretty fun to watch it, just because within like the first 20 seconds, like all the aggregates just started to break apart and the bowl was looking pretty brown by the end of it. But this is actually meaningful because, you know, when the soil becomes saturated, like it does all the time, you know, when it rains, whenever, you know, if these large pore spaces, you know, if they're stable, then the water, you know, will move freely without causing the aggregates to break apart. Oh, right. So just finishing up here, like, so the Slake test then has been considered a useful proxy, which can sort of maybe indirectly provide information then about the presence and quality of organic carbon in the soil, which then can be compared to other measures. So as I just mentioned on our course, we did a few experiments comparing different land uses. So as well as conducting a Slake test, we also measured and estimated soil organic matter and carbon. And this was via the loss on ignition method, which is a commonly used combustion method to measure soil carbon. And so this chart here, this just shows the mean concentrations of soil organic matter and carbon stocks. So these yellow blocks here, this is our soil carbon. And so you can see like the much lower levels of soil carbon here in the arable plot, So around 20, we estimated it was about 23 tons per hectare. This is quite similar to a lot of agricultural sort of soil carbon stocks. And this, but you know, ultimately, this translates really well to the stake test, right? Because although the stake test, you know, it was our subjective sort of interpretation that we use as scoring criteria, you know, we could clearly see the difference here where we scored it a 3. So going back here, 3 was like, you know, over half of the structural integrity was lost in like the first 30 seconds or whatever. So, you know, and this really translates to, you know, the lowest soil carbon stocks here. So we could, you know, these experiments were perfect, by the way. There is no way this is going to be entering in some scientific paper or published anytime soon. But what they did is they really helped us see this clear relationship between aggregate stability and soil carbon, which is reflected in a lot of other studies. And, you know, like, this makes sense because the presence of carbon in the soil, this is what's ultimately influencing the binding agents which hold these soil particles and the aggregates together. And that's about it, really. I hope that I've blown your mind enough with examples of all the intricacies behind soil carbon. And yeah, I hope it's been interesting and useful. And yeah, I've got some references here. I didn't actually manage to squeeze all of them on, but I'm happy to paste all the references that sort of, I think I've probably read over like 30 to 40 scientific papers over the past half a year on carbon. So I'm happy to share anything with you guys on the network. But yeah.
Thank you, Mary. That was so amazing. Because considering, was that one of your first presentations?
My first like official kind of presentation ever, I guess. Like, I don't know. Yeah, I've done presentations kind of at uni, but this is, yeah.
Oh, wow. That was just, you nailed it. It's really great.
Oh.
I loved what you said about like the best way to learn is to teach. And I thought so often through that, it's just like, oh, I really like Mary to be my soil teacher.
Oh, wow, that's a massive compliment.
It was brilliant. Really, really great. Thank you. I'm going to reclaim the home.
Oh, yeah, do it.
Yeah, but that's one of the hard things about doing talks on Zoom is like that we just recorded that was just your screen, so you couldn't see, you know, you can't like read the room, but you read it really well. Like I thought your expression was just you had it. For me anyway, I was completely with you. So yeah, just well done. Thank you. Really great presentation. Cool. Well, it's hard to know where to open up the conversation there. There's so much. But we got some amazing people in the room. Like really, I mean, yeah, I don't know where to start. We've got Eddie, we've got Brian, we've got Josh, we've got Amy, we've got, but I mean, like maybe we could, give the new folks a chance to say hello. Would that be a good place to start? And just introduce themselves, and then we can sort of find topics that might naturally occur, because where Mary's talk was so sort of expansive, it might be quite hard to just be like, okay, let's just go in there. Hey Josh, welcome.
Right.
Yeah.
Great talk. Cheers for that.
Excellent. Yeah, wicked. Josh, you're doing some wicked stuff in Devon with agroforestry and Kashi. Martin was singing your praises early days, and yeah, we're really glad to see you here.
Oh,
so
right.
what were you up to recently?
Yeah, so my name's Josh, and I basically manage a market garden in North Devon, about 17 acres. But it started from scratch. We've basically taken a dairy field, so we have really intensive dairy here, like, you know, we look like 12 silage cuts and constant slurry every single couple of weeks. So it's pretty intensive, quite damaging. So our field used to be that, and now we're turning it into kind of, I don't know, an agroecology or regenerative, or however you want to put it, kind of style of growing. But our main emphasis is on agroforestry. So it's quite heavily kind of edible forests and syntropic and So like we have like a 10-acre agricultural edible forest where it's like an edible forest with an efficient pick, like Volta Van Eck in Holland. And then we have like a romantic edible forest, and we have some tropic grows, and really we're finding ourselves kind of like reducing our annual vegetables each year, and trying to go more of a perennial based system, and alter the perception of like, should our diet contain 90% annual vegetables, or should it contain a higher percentage of perennial crops instead? And for me, I studied in Japan. I did some farming in Japan, and so like natural farming is quite close to my heart. So like the annual growing that we do, we try and base it on natural farming or Japanese natural farming, to maximize ecology, you know, as much as possible. And that's where like the cashew comes into play, and all sorts of different things like that, really, because it kind of suits what we're trying to do.
Thank you, Josh. Yeah, so, I'm super stoked that Josh is gonna give us a presentation and another conversation next month. So on the 21st of June, we're gonna hear from Josh in depth about what he's up to, which is really cool. Nice one. And Eddie, how are you doing, man? It's good to see you there.
Thank you so much. I'm really happy to be here. I have to admit, there's no way that I could have got onto this without, well, Muriel's help there. And thank you so much, Thomas, for your help getting us here. That's amazing. Thank you so much. And I'm sure, Mary, I've got lots of questions, but it's all swimming about in my head at the moment. And
Same.
so, I mean, can I ask one simple question? Though, I mean, you're talking. There was that last, you know, thing came up, and it was comparing the aggregate stability in woodland, grassland, and agriculture.
Yeah.
Yeah. So that was really fascinating for me, and I ended up just wondering, you know, like where the cut-off point between a grassland and a woodland actually is, you know, because I'm thinking about like my favorite bit of land, which is really near here, and it's a piece of land which was you know, intensively and exploitively farmed, but it was bought up by the water company because it's surrounding a sensitive bit of water. So they bought it as a buffer zone, and they just left it alone. So so it's had a transition from some really intensive like agricultural land to grassland, and slowly to woodland, I suppose. But it's just, the transition is incredibly slow. And, you know, looking at it at this stage, in the last decade, you know, it's more shrubs really than trees, you know, and they're beginning to assume dominance over really what it's like moss and really kind of like tough sparse grass, you know. So
Yeah.
is it, you know, so a question is like, is it best to just leave that land like that alone entirely, or is there some treatment you can give it, or what do you do, you know?
Oh man, this is where I struggle with it, because I'm learning like all the background to it, but in terms of actually like what we should do, because I think a lot of this also comes down to precision, and it depends where you are and the stuff you've got there. Like I find it quite difficult at this stage in my pathway to be able to like advise between that, especially when we're comparing, because I do a lot of comparisons, sort of learning, like you say, between like agriculture and compare like, oh, this bit is, you know, intensely managed agriculture compared to that. But if we're thinking in terms of like grassland versus woodland kind of restoration, you know, I mean, I am heading down this path, you know, of like just leave it. It depends what your mission is, because the problem with this is like, I'm doing this presentation because we're all really obsessed with carbon, right? But like a lot of places of our land, maybe we don't need to be so intensely being like focusing on carbon and sequestering all that carbon there. Like it depends. If your ultimate goal is like, hey, I've got this bit of area and I want to sequester loads of carbon, then yes, maybe I would think a bit more about maybe trying to have some input there. But if that's not so much of your focus, I would actually just maybe just let it do its thing.
I loved what you said, Mary, like microbial restoration. Like I've never put those two words together. Like I put like,
it's so new. It's so new.
Restoration, but that is a complete, almost, it feels like a completely new term. It's like it's got its own value, hasn't it? Adding the microbes, which just their effects are more immeasurable to us at the moment than like a static mineral like carbon. We've got Brian in the room here. Brian is a fascinating biochar sort of stove innovator, and yeah, he and I have done some work with easy ways we can make biochar. But I mean, that's inorganic. That's the mineral carbon, right? Like the stable carbon, which is supposed to be less in as part of the cycle that you described, Mary. But that can definitely have some pretty profound effects on creating, increasing soil fungi and things like that. At least there's a lot of studies about that. But yeah, I mean, what you were describing there, Eddie, it's very much a landscape in succession, and that succession, you say now it's covered in shrubs. That's the work of the trees constantly pumping their exudates down and feeding the fungi and to allow more woody perennial forms of life to emerge, and eventually it will end up being a patchwork of forest and meadow, like most of this country was before we sort of started managing it so much intensively. But yeah, it does.
Okay, so a couple more questions coming to mind, but I'm also, like you say, there's some fascinating faces here in front of me. I'd love to hear what other people have got to say as well. So yeah, well, hold those thoughts. Let's any other sort of burning questions from Mary's talk that people just want to throw out there, and we can try and cover. Yes, Peter.
It's not a question really. It was something which Mary said, which had never occurred to me really, that with soil drying up and getting wet, you've got soils which are going, vacillating between aerobic and non-aerobic, which I've never really thought of therefore, because I've been thinking quite a lot. Mary, we collect food waste and create gorgeous compost. But one of the things I've been looking at is, we're about to run an experiment of basically having two large wooden coffins. So post hot composting, having two different bins sitting there, one of which will keep relatively aerobic, but the other will let seal. But the one which is let seal, I'm going to, with Martin's help, bokashi into it. So trying to create a different world of life in that way. And then compare the two in 3 or 4 months' time. But I hadn't thought of it in that way of, lots of soils get wet and dry all the time. We have access to a bit of land, and one of the things that we were going to put a pond on it, but what the people said was, the advice is don't put ponds, put scrapes. So essentially make scrapes which will get wet and dry, because that will be better for life than having a pond, which is one thing, but having change, I suppose, and edges. So anyway, there's lots of stuff that you were talking about, but I think that's particularly fascinating for me, that thought that things get, and within the composting process that I have now, even in the hot composters we definitely have a vacillation between aerobic and non-aerobic. But up till now, I've always pushed towards aerobic, I think, as being the ultimate goal, to get as much air in as possible in quite a traditional way, whereas Martin's bokashi work is that's not what that's about. And we've now got a bit of bokashi experimenting going on as well. Anyway, that was just particularly, there's a lot you said. Thank you so much for that. Bloody hell, too much.
I know.
Oh, blimey. No, it's not too much. It's fantastic. And although the final reflection is, it's like, I just hope there's loads and loads of universities and places all over the world who are looking at this. It feels like humankind is catching up horribly late and all this stuff. Some of what you're talking about, it feels like this is what we should have been being taught you know, in school decades ago. It feels so important, but yet we're coming to it. It's, you know, horribly late in the survival or not survival of life on Earth. Anyway, that'll do.
Yeah, I think that that's a really good point. I think definitely we're quite late on the biology side. We all know that. Like, we're quite late on being like, hey, the soil has biology in it, guys. Do you know that? But I would say that, because I sort of felt like this when I started, I was like, why were you just doing this now? Like, but you know, when I was talking about the paradigm shifts, like, a lot of this is being driven by advances in scientific technology. So things that we didn't actually have before, the capacity we didn't actually have before. So, you know, from that perspective, that is kind of why we're catching up a bit late, as it were. But that's also that's a really interesting thing you said about scrapes. I'm going to write that down, because I sort of told you about this, because I found that this, because I was like, when I started learning about this, I was like, oh wow, this is cool. You know, carbon can bind to minerals, great, and they just stay there forever and whatever. But then when I found out about this cycle of aerobic and anaerobic, I was like, oh, okay, well, that puts a bit of a spanner in the works, doesn't it then? So,
Yeah.
but I haven't really thought any further about how, you know, how does that translate into land management? So that's quite interesting what you said about scrapes. Maybe that has some influence.
Yeah, no, it's interesting. Actually, there's a couple of things. Look, you see, I've just picked up this book here, and it's called The Zinc Solution, and it's written by the guy who alerted us to the problem of combustible lead in petrol. Unfortunately, he's dead now, but it's like, well, it's like, probably, you know, it's all about trace elements and zinc in particular, and the damage it did to, you know, people, and it's still doing now, the absence of it. So the thing is, we it's not that we want these trace elements or carbon to be locked in, but to actually be moving through a human system and out again and back into the soil, you know, because like the incredible depletion of trace elements in agricultural soil at the moment is not ever going to be something that's easy to repair. And, you know, basically the way that we have a broken sewage system at the moment is not helping in the least, you know. So just including ourselves in this like living system is an essential way of thinking now, because, you know, like there's so much repair work that really needs to be done, you know. So anyway, I certainly recommend that book, because it's a bit of a history book now, but it's the thinking, and the guy that was doing some incredibly careful thinking at the end of the last century there. And what's his name? It's Professor Derek Bryan Smythe and Liz Hodgkins, and she's still alive. And I've actually sent her a message and say that I want to chat with her. So,
you, Eddie. I just want to quickly connect you and Peter just quickly as two pretty local mycelium compost makers.
Okay.
So Peter is Luke Froome, and Eddie, I remember chatting to you at the Green Fair, and you were saying you were, you know,
near Winchester,
reached out already, tried to reach Peter. Well, there he is, and you're not far away. You could both collaborate in local composting. So there.
Okay. And let me write your name down here, and then. Or put your email address in the
contact. Don't worry about that. You'll have his contact through the forum, which is now.
Yeah, of course. Yeah,
just contact him there. But then I just wanted to say, thanks for sharing that about zinc. I've totally heard that's a really deficient mineral in a lot of diets, and I wanted to just share, because I've just been watching some more Matt Powers, like regenerative soil stuff. He says at the beginning of one of his sections that carbon is very much could be seen as the stage upon which all of the other mineral cycles kind of play out, which I thought was a really good interesting way of framing it, because all of the minerals are so vital. We've got to get them into all of our cells, and all animals do, and we'll have an instincts as how to do that. But I guess the instincts have been sort of washed aside now with like the scale of which our economic system has been doing everything. Compost is definitely one way we can start enabling those minerals to sort of cycle properly again. And yeah, I just wanted to maybe invite Martin to sort of say something now, because we've got Martin here, who's an amazing, Bokashi teacher, and sort of facultative microbe spokesperson, which is the facultative microbes are the ones that operate between the aerobic and the anaerobic states, and as Mary kind of alluded, and therefore the most potentially successful or desirable in a lot of soils because of the wetting and the drying processes that
To be honest, Tom, and one thing I want to just compliment Mary on, never ever thought of it like that in respect to the soil system. And actually, it's a very simple way of looking at it, but if for the newbies, etc. My name is Martin Richards. I work for a company called Aggregates, and we sell effective microorganism products by a guy called Dr. Higa. And we have a great team with us that involves anyone and everyone in respects to sharing data and sharing the network. And I joined the Mycelium network to try and gain my own knowledge as well as share some of my own knowledge. And when I first started talking to Tom, we started obviously talking about the anaerobic systems and the microbes that are involved in that system. But one of the things to bear in mind with all these systems, the same as soil, is microbes can work in a facultative system, i.e. they can work in anaerobic and aerobic, and depending on what your output wants to be, depends on obviously what you're trying to gain. But I think microbial restoration, love that. I think that's a very good way of kind of putting microbes back into that system to be able to then develop their own microbiomes in the soil. But also, we sometimes when we look at, and I know quite a lot of this network was set up from composting and the food revolution and trying to close the loop in respects to that process. But we sometimes forget that the most important part of that process is the soil, and that's what creates the structure to be able to do that circular economy. But we rely so much on an input and then an output, i.e. a plant being grown and then obviously the output being a yield, that sometimes we forget that actually, if we don't think about the soil as a system and the biology in that system, then unfortunately over a period of time you will get disease and you will get issues with yielding, etc. And I think if we look at the people in this room, we all have kind of sort of parts of that. I will bring Josh into this conversation because I know he's smart as well. Is that Josh, as a person in respects to what he's developed, is really looking at that circle as a system, because not just from a business point of view, but from a personal point of view as well. And one thing that he's taught me is that the best way is diversity. The best way for us to work together is that diversity in respects to that soil system. And what Mary was saying about carbon, do you know what, I agree with you. I really do agree in respects to carbon is not the be all and end all. I spend most of my time with businesses talking about carbon credits, biodiversity credits, which are going to come in November, kind of selling what's in the soil. But actually it doesn't come down to that, because as humans we don't rely on just carbohydrates in our diet. We need proteins, we need fats, we need carbohydrates, but we also need, and one of the most important parts for to keep ourselves alive for a longer period of time, is vitamins and minerals. These are sub parts of this system. So when we think about how we're going to put those back into the soil, without that diversity in the soil in the first place, the microbiology cannot process these minerals into usable, and that's the key thing, usable nutrients in that cycle. So farmers unfortunately have got into a bit of a cycle of chucking things at the plants. Nitrogen is a good example of that, and unfortunately the output has not been as good in respects to yield, and they kind of got stuck in this kind of loop. The ones that are going to do and make a change in the future are the ones that are looking at kind of nutrient values of a carrot. And in my perfect scenario, I want to go into a supermarket and go, I can buy this one carrot which is an A+ rating, or these 10 carrots which is a C rating, and actually the value in that nutritional value is that one carrot has the same nutritional value as those 10 carrots in respects to how they're buying. But unfortunately, societies, it's very difficult to get to that point. But yeah, that's my penny worth.
Yeah, no, thanks. That's really interesting, Martin. Yeah, we've lost that nuance with food, don't we? Plants are so resilient. They can produce they can do their thing, but just without any of the mineral integrity that they're supposed to have. And it's so hard to teach people that, to value the more expensive food. And yeah, super complex. That's another thing. Okay, so I just want to first of all, everyone hold their thoughts and questions. I have to share one thing with you guys before potentially we all have to go separate ways, which is simply a feature of the network, which I've realized is not very, very well communicated, explained maybe. And it's basically just how to post events, because I realized that one of the things that this forum could be really useful for is us getting more of these events. We do our monthly events on them, and some of our community, like Nikki and others, have put up other events that are going on. But I went to put up an event the other day and I was confused. All we need to do is know how to do it. So the reason it's a little bit confusing is because of this new thing called Spaces. So Spaces appeared a couple of months ago, and now we've got all of these spaces, which everyone can join and is joining, and they're sort of to go deep into those different areas and support each other and ask the questions. But events can be more general, and there's a place for everything that's general, and it's the home feed. So when you want to create an event, this button create is always in the top corner, but you click on it and it comes up with all the spaces, and you're like, I don't want to just create it for one of the spaces. I want everyone to know about my event. So you have to click the home page. And then when you click the home page, you can decide if you want to go live, do a quick post, write an article, do a question or a poll to everyone, or organize an event. And you click on the event, and then you've got the normal kind of thing, just like any Zoom event. You can decide what to do, when it is, etc. And it will post it like these things. Okay. Hope that was helpful. And if we could also, if I just really think this could be one of the most useful things the forum could facilitate, as well as the spaces, is an event count page of what's going on around the country.
Tom, can I just butt in? If because I really want people to use this, because it's really interesting for me to know what's going on in the UK, and hopefully maybe get out of the office a bit more often. Anyone that puts stuff onto this homepage, really happy to put on my socials in respect to the Action group. So if you want a bit more exposure, etc., I can do that. But I don't. If you can add it to this, and then tag me into it, and then I'll put it onto our socials.
That's really cool. Likewise. Yeah, that's cool. Thanks for sharing that, Martin. Yeah, so other than that, it would just be really good for all of you, Josh, if you could join the Bokashi Biofertilizers group. Have we got an agroforestry? We should definitely have an agroforestry space. Likewise, just suggest stuff. It's an emergent thing. Like, I don't have the answers. I just put these things because that's what I think is really important. But there could be more. It's just, like Matt asked some really good questions recently about drink fermentation. So I made a space for that. Like, it's all working with microbes. It's kind of all. I'm quite into the sort of expansive definition of composting. It's just partnering with microbiology in my eyes. So we can have these focus spaces. No one else has to, be exposed to it if they're not interested. But if you are, go and find the people that are interested in. It's gonna be useful. Cool. Back to the soil carbon. Yeah. Right. Well, what questions have I got? Has anyone else got any more questions, or are we kind of feeling like our brains need to process everything.
Yeah, yeah. I got a question. It's something that I would really appreciate this group here helping me to think about.
Yeah.
And you know, it's like when I'm working the soil, I'm always coming across bits of plastic, you know, which is obviously carbon. And my point of view on it previously has been, well, that's got to come out and go into the bin. But also at the back of my mind, it's like, well, that's going to go into an incinerator and be really, really dangerous for somebody's kids, you know. So, you know, rather than hating it, I'm also hearing all these things. Well, there's these, you know, funguses which are developing the ability to save us from plastic, you know, and it's sort of like sensational headlines and everything like that. So, I mean, actually right at the moment, I find myself very ambivalent on it. And I find myself looking at pieces of plastic that I picked up, and some of them seem to be so incredibly weathered and, you know, sort of like rich in organisms anyway, that I just chuck them into the compost bucket, you know, to be recomposted again, you know. So whether that's right or not, you know, that this is the kind of ambivalent place that I find myself in at the moment. What do people think on that one?
That is an interesting comment. Yeah, I mean, I have seen that there have been studies published on like kefir, which is mainly lactic acid bacteria, the same stuff as in bokashi. Kefir has been proven to break down microplastics.
Okay.
So I mean,
No,
I think this is an enormously like understudied field. I know obviously there's the oyster mushrooms that can break down polyurethane. But I try and pull out all plastic apart from the home compostable ones in all of the composting I do, just because I don't think it's helpful to soil organisms. I've also seen like nematodes are getting it's killing nematodes. It's getting stuck in their intestines and they're dying.
Right, microplastics.
Microorganisms that get microplastics, exactly. I definitely don't think it's good for animal and plant bodies. Yeah, any other takes on that? Like, I mean, that's really all. I don't have loads of studies or anything to
and
report.
the impact on earthworms.
Earthworms, exactly, yeah.
Right, it's damaging to earthworms.
Yeah, studies prove
Yeah.
really,
I would
really.
strongly not recommend. I know I've talked to some people and they're in disagreements about, you know, their partner, whatever, chucks the Hoover bag into the compost heap. I do not think that is a good idea. We mainly Hoover microplastics up off carpets, and like it's really quite important that we say to people that's not good. That's not compostable material. Microplastics do harm to microbial and animal bodies. I think that's yeah, I can't see how that isn't the case.
Yeah.
And then again, eventually, you know, I guess the whole biosphere is going to filter up the microplastics and create new life, and it was just how much harm will that cause, and that's what we don't really
Well, I mean, you know, sort of a microplastic is a piece of plastic which is broken down, and then sort of like, do microplastics then break down further? And how small do they have to get before they just actually, you know, become elemental carbon again? Or does that never happen?
It's a big question. That's a big question. We should get some scientists that have done a bit more research, like looking into this and get
Well, if there is any.
Yeah, that is huge. Yes. Hi.
I need to go, but I just wanted to quickly say thanks to Mary before
Definitely.
for making it feel accessible. It's like really complicated stuff, and it's just I've really, really appreciated your posts also, Mary. So thank you for that, and see you all soon.
Oh,
Yeah.
cheers, Amy.
Okay,
Big love.
Yeah.
see you. Bye.
Awesome. Oh, wicked. That could be a good note to end it on. Just thanking Mary for like a really
Brilliant. A brilliant presentation.
Oh, thanks, guys.
Yeah, Mary, really good. Really, really good. Thank you. That's awesome. Yeah. it's one of those things where like you just have so much to ask, but also you just don't quite know where to.
Yeah.
And we don't have the time to really answer many questions because they're so.
To be fair, like if anyone, because honestly, my brain is, I've literally been like non-stop for like the past few months, and my brain is, you know, I probably don't have that much capacity now anymore. But if you guys actually do think of any questions, like just post them on the chat or like anywhere, and yeah, I'd be happy to like answer anything if you
Thanks.
think of anything.
Yeah,
Great.
Martin, I hear you. We're gonna make a map, and we're gonna like put all of the little dots on of where everyone is, because yeah, that would be really good. Definitely. And the references, yeah, Mary, would you mind doing a post of your reference slide on the forum?
Yeah, I think what I'll do is I'll do a post and I'll put my references on there. And actually, if anyone has any questions, maybe ask them. I'll put any questions below, and we can keep, so it can stay on the forum instead of getting like lost in the WhatsApp chat or whatever.
Will it go in the mycology section, Mary, or just on the home feed? Maybe just on the home feed.
Yeah, it's not really strictly mycology. It's quite, I'll keep it on the home feed.
Yeah, okay, great, perfect. Awesome. Well, thanks everyone for tuning in. It was really
Thanks, Tom.
great meeting. Yeah, brilliant.
Thank
All
you,
right.
Mary. That was great.
Have a good evening, guys. Thank you.
Okay, cheers everyone. Bye.
Night.